Blood. Blood is a connective tissue with a liquid matrix. Just like all the other connective tissues, it has cells, it has proteins. In this case, the proteins are not in the form of fibers, but they are suspended in the fluid, and the matrix, which is liquid. Um, Blood has quite a lot of functions. The obvious one is the transportation. It transports gases, oxygen and carbon dioxide. It transports nutrients like glucose. It transports waste products, um, let's say to the kidney. Uh, so it is the major method for transporting different things from one place in the body to another. Uh, some substances need to be processed in a particular place in the body, and the substances that are precursors may be produced in a different part of the body. So the blood is going to move those parts to a new location where they can be modified, turned into something that the body needs. So, for instance, the precursor to vitamin D is produced in the skin, and then it's transported by the blood to the liver and then to the kidneys to process it into active vitamin D, and then the vitamin D can be transported to the intestine, where it promotes the uptake of calcium. So there are various examples of that. It transports regulatory molecules, which means hormones and enzymes. So we just finished the endocrine system, and you know that the definition of a hormone is a signaling molecule that is transported by blood from one part of the body to another, from an endocrine gland to wherever the target tissue may be. There are other things that blood does apart from transportation. One of them is the regulation of pH. Uh, it helps. It contains buffers, and buffers keep blood pH within its normal limits. And you need to know those normal limits: 735 to 745. Uh, so blood is going to regulate the pH. If the pH of blood is well regulated by buffer, then the pH of extracellular and intracellular liquid is also going to be regulated. Um, Blood also is involved in the maintenance of body temperature, and in the another video that I already posted, there is quite a lot of discussion on how uh, body temperature is regulated by blood flow. But there are other ways. So blood flows through areas that are metabolically active, so it gets heated, and then it's transported to the surface where the heat is released from the blood. So body can regulate how much heat to lose by opening and closing the capillaries in the skin, for instance. Um, Blood is also protecting us against foreign invaders. So there are types of cells in the blood and different types of molecules in the blood that are part of the immune system, the defense system that protects us against, pathogens, viruses, bacteria, uh, uh, toxins, parasites. And when blood vessels are damaged, blood has clotting factors that are going to help heal the wounds in the blood vessels, close them so blood is not lost. So it has a role in repair of blood vessels and bringing them back to their normal function. So there's a lot of things that the blood does. So, as I said, the blood is connective tissue, and the liquid matrix is called the plasma, blood plasma, and the plasma is only about 55% of the volume of the blood. The remaining 45% are formed elements. Those are cells and cell fragments. Uh, the blood volume in an average female, adult female, is about 4 to 5 liters, and about 5 to 6 in an average male. So a little bit more in men because men tend to be bigger. Average male is a little bit bigger than an average female, so we'll have a little bit greater volume of blood. Blood is about 8% of total body weight. So when we look at the blood plasma, uh, 91% of it is water, and about 2% are various other solutes, ions, some nutrients, and waste products, gases, hormones, things like that. So very small. But 7% are proteins. And proteins in blood, in plasma, come in three flavors. First are albumins, then globulins, and fibrinogen. Albumins are carrier molecules. They attach various other molecules that need to be transported from one place in the body to another and carry them there. They also add to the osmotic pressure of blood, regulating the exchange of liquids between the blood and the extracellular and intracellular liquids by drawing water towards itself. Globulins, some of them are carriers, and the others are part of the immune system, antibodies. And fibrinogen is a protein that is involved in blood clotting. So we are going to talk a little bit more about blood clotting later. So let's now look at the formed elements, those 45% of the blood that are cells or cell fragments. So the most numerous ones, and you've seen the slides in the lab, are the red blood cells or erythrocytes. And there's, millions of them, 4 to 7 million of erythrocytes in an organism at any given time. Uh, then there are white blood cells, which come in different varieties: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And then there are platelets. Platelets are not cells; they are cell fragments, and they are involved in blood clotting. So those are kind of main components of blood. So when we look at the formed elements, when we look at the cells, let's look at them in a little bit more detail. So we have red blood cells, we have different types of white blood cells, and we have platelets. So red blood cells are most common, yet they are part of the same developmental system as several other types of cells. Hemopoiesis or hematopoiesis is the process of development of various blood cells out of its stem cell. Hematopoietic stem cell is called hemocytoblast. Hemocytoblast can develop, which means you can have cell differentiation. When you start with a stem cell, it's going to differentiate into something else. And it's going to differentiate in two different ways. It can become a myeloid stem cell or it can become a lymphoid stem cell. Myeloid stem cells can become all sorts of different things. They can become, then, as cell differentiation continues, they can become red blood cells. They can become various types of white blood cells like neutrophils, eosinophils, basophils, and monocytes. Or they can become megakaryocytes. Megakaryocyte is the precursor of the platelets. That's the cell with lots of nuclei that breaks up into fragments, and each fragment is a platelet. On the other hand, the lymphoid stem cell as the cell differentiation continues, can become a lymphocyte, which can be a T lymphocyte or a B lymphocyte. And we're going to talk much more in greater detail about those two when we talk about the immune system, as well as NK cells. Those are natural killer cells. They are part of the lymphoid stem cell developmental pathway. So red blood cells are you've seen them under the microscope. You've seen pictures of them. They're small. They look like discs. The edges are thicker than the center, so it's like concave discs. And you may have remembered that the red blood cells don't contain a nucleus. Uh, that is true for most mammals, but not all of them. Red blood cells has nuclei in animals like camels. Also, red blood cells have nuclei in birds. But in us, in humans, and this is human anatomy, uh, uh, our red blood cells do not contain a nucleus. So, what is a red blood cell? Sure, it's going to have some RNAs and proteins that can continue doing some stuff, but you don't have a nucleus, so you don't have the DNA, you don't have the instruction manual for the metabolism of the cell, which is why they don't last very long. What red blood cell essentially is packaging for hemoglobin. And I have already posted an entire video about hemoglobin. So, red blood cells are packages that deliver hemoglobin to various parts of the body, to the lungs, where hemoglobin uh, is going to bind more oxygen and is going to release carbon dioxide that is attached to hemoglobin. As I said in that video, only a small proportion of carbon dioxide is actually bound to the hemoglobin. More of it is going to be transported in other ways in the plasma. And then hemoglobin goes to all the other parts of the body, which are going to take that oxygen from hemoglobin, and they are going to put carbon dioxide in the blood. So red blood cells are carriers of gases, of oxygen and carbon dioxide. So that is the primary function of the red blood cell. So I have given you an entire video about hemoglobin. So watch that. I just want to say something more about the fate of hemoglobin and of red blood cells once that red blood cell, without DNA, without a nucleus, gets to the end of its useful life. So, what is going to happen there is that it is going to be broken down, and and hemoglobin is going to be broken down too. So, globin part, the alpha and beta chains, the protein part, is going to be broken down into amino acids, and those amino acids are going to be reused for building other proteins, including more copies of hemoglobin. Uh, Iron is going to be recycled. Iron is going to be almost entirely recycled. And it's mostly going to happen, that recycling is mostly going to happen in red bone marrow. And this is where red blood cells are made. So, this is where all that iron is going to be used for producing new hemoglobin. A little bit of hemoglobin, a little bit of iron may be lost in urine or feces. So we need to take in some iron in our food, but most of it is wonderfully recycled. So what we are left with is the molecule of heme. Remember, there are four molecules of heme attached to the molecule of hemoglobin, and that one is not recycled. That one is broken down, and the breakdown product of hemoglobin is called bilirubin. Bilirubin is actually toxic, so we need to get rid of it. And most of it is going to be uh, released by the liver into as bile into the uh, into the gallbladder or directly into duodenum. We are going to talk about it when we talk about the digestive system later in the semester, and so that bilirubin is going to be part of bile that is going to be secreted into the inside lumen of the gastrointestinal tract, and there most of the bilirubin is going to be just go through the whole GI tract and go out with the feces. Bilirubin is a pigment. It's yellowish, but bacteria and enzymes inside of the GI tract are going to modify bilirubin, and so this is what is going to give our feces the brown color. uh, a little bit of bilirubin actually gets modified inside of the intestine and gets absorbed. Uh, and if it gets absorbed too much because something's not working, let's say bile is not working, we get jaundice. But if it's modified first uh, uh, and then absorbed, those are going to be different pigments. They're not going to be brown; they're going to be yellowish. So they're going to end up in the bloodstream. They're going to end up in blood. Once that blood passes through the kidney, those modified molecules of bilirubin are going to be excreted in the urine, and there they're going to give the urine its yellow color. Okay, when there's a damage in the blood vessel, the blood can leak right into other tissues and mess with the function there. Or it can be external bleeding, which means we are losing precious uh, liquid, precious water, and proteins and cells through that bleeding. So we can't tolerate more than a small amount of blood loss. I mean, we can produce new blood to replace it, but it takes some time. So, large blood loss is going to lead to death. So, our bodies have evolved mechanisms for minimizing blood loss by quickly repairing the damaged vessels. And that is a process that has three steps. The first is vascular spasm, the second is platelet plug formation, and the third is blood clotting. So vascular spasm is immediate response. It's a vasoconstriction. It's a constriction of that blood vessel. So much less blood or even no blood goes through that vessel. The less it goes through it, less there is to be lost through the cut or opening in the wall of that vessel. So smooth muscles that are surrounding the wall of that blood vessel are going to contract, constrict it, so they can either completely or partially uh, stop the passage of blood through it. There are all sorts of chemicals that stimulate vascular spasm. They are going to be uh, the paracrine, essentially, signaling molecules that come out of the damaged blood vessel wall, as well as by platelets. So that's the first thing. So you contract that vessel. It's instant, it's quick response to immediately lower or stop the blood loss, but it can't last forever. So you need to do something else. You need to actually repair it. So the platelet plug is the next thing that happens. That's the accumulation of platelets. Remember, the platelets are tiny little fragments of cells, cytoplasmal membrane, and they can gunk up that hole. They can seal it up. They can fill the hole with themselves and thus produce kind of a temporary wall of that blood vessel before it is regrown. So plug formation is second. And finally is blood clotting. Blood clotting or coagulation is results in the formation of a clot. And the clot is made of fibers, and those fibers are fibrin fibers. and those fibrin fibers are going to trap in their mesh blood cells and platelets and fluid. So, in order to form a blood clot, we have to have a complex series of biochemical reactions, and those are clotting factors, a number of them, and they normally just sit in the blood and don't do anything and do not cause clotting. But when there's an injury, the clotting factors are activated, and it's a complex process and it's a chain reaction. So the clotting factor, I'm going to simplify it. Clotting factor one is going to stimulate number two, is going to stimulate number three, is going to stimulate number four until you have like a dozen of them all stimulated. But remember, we mentioned earlier in the course that this is a positive feedback loop. They're also having all those clotting factors are not just stimulating the next one, they're also stimulating the previous one. So clotting factor number three is also stimulating the one and two. So you have more and more and more, and it really expands really, really fast until they're very, very activated and they don't stop until the goal is reached, and the goal is the formation of the clot. So once the clot is done, it's made, it's in place, then all of that stops, just like every other positive feedback loop. Okay. We, you have probably heard, and we're going to talk more in the lab about ABO blood types. So, in cases when there's a need for transfusion because of blood loss or something like that, we have to transfuse the blood of a compatible person. Compatible person is somebody whose blood we can take without a reaction. It's not going to be an immune reaction, an antibody reaction that is going to destroy the transfused blood red blood cells of the transfused blood, and that would also lead to all sorts of other things that go with the immune response, like inflammation. So, how does this happen? Um there are uh, molecules on the surface of red blood cells. Uh, that can be molecule A, or molecule B, or you can have both, or you can have none. If you have none, you have an O or zero. If you have only A, it's A. If you have only B on the surface of your red blood cells, you have blood type B. If you have both, you have AB. So if you have A, for instance, on your uh, surface, that also means that you're not producing antibodies against A, because you would then destroy your own red blood cells. But that also means that you probably have antibodies against B. So if the red blood cells that are coming in through transfusion have the molecule B on their surfaces, your antibodies are going to destroy those cells. And now you have suddenly a carnage of millions of red blood cells destroyed, falling apart, leaking, all sorts of things, hemoglobin uh, into your bloodstream. That's not a good idea. You're going to die probably. So that's why we have to do the matching. So, whatever is your blood type means that you have to take in the blood that is compatible. The blood that is compatible is the one that has red blood cells with the molecules on their surfaces to which you do not produce antibodies. So if you have A, you can take in A. If you have B, you can take in B. If you have AB, you can take AB. If you have zero, you can take zero or O. But there are other combinations, right? So uh, the O is universal donor, right? You can give uh, blood O to anybody, to any recipient, because there's nothing on the surfaces of the red blood cells. There's no A, there's no B. So no matter what antibodies you might have, A or B or both or none, doesn't matter. You can receive 0. On the other hand, AB is a universal recipient. If you have AB, you can take whatever blood you want, right? Because you are not producing the antibodies against A or B, so you can take any blood you want. Um, so you need to know those tables, what can go with what. Uh, and RH factor is similar, goes a little bit different than ABO, and we are not going to go into the details of that. And I'm going to end with blood here. Please also watch the other video that is specifically about hemoglobin.